So I'll be talking about my package, BioSimulator.jl. It is a package for stochastic simulation in Julia. And if you'd like to look at slides or check out the package, just be mindful that you can get this at this URL here, but be mindful that my package isn't registered in Julia, so you do need to install that manually. So as we saw in the previous talk, stochastic, complex biological systems, these networks are really complex, as you can see here. So my job as a biomathematician is to help scientists make some meaningful progress on these complex problems using the computational tools that I have at my disposal. So I'm going to start a little bit backwards, introducing the types of problems that I work on by talking about the simulation aspect first. So often a master equation is a useful starting point in writing down stochastic models. And the reason is that it allows you to focus writing down rules for your system, how things behave, rather than thinking about the underlying distribution for things, worrying so much about notation as you see here. I'm not going to go too far into details of the math, but you can go ahead and look at this on your own if you'd like. But essentially what simulation does is we sample the master equation, and the reason that we do this is because this almost harmless looking equation actually represents possibly countably many differential equations that are coupled together. And while there are many tools in mathematics to deal with these sorts of things, sometimes we just want to test simple ideas before we commit to doing any sort of analytics or asymptotic results on these things. And sometimes we just don't have the math to actually solve these things. So some examples of how we translate these rules to translate the mathematical description to rules are on here. You can think of one reaction as or an event called budding. You can think of it as a birth event as well. The idea is that you have some species X and some event takes place, and all of a sudden you have two X's instead of just the one. And these things occur with some sort of intensity or propensity. These reactions have their own internal firing times, and these firing times are somehow weighted by these intensities. So while the math, essentially at the end of the day, we just want to help scientists make progress on these problems. And one way that we can do that is by mapping biological problems onto something that we know how to solve in math. So why stochastics? Stochastics just means random noise or some sort of process that you don't really understand underlying your the deterministic features in your model. You can think of balancing between directional forces in your problem, such as you can think of like velocity in some sort of equation versus non-directional forces like noise. But we do have to be careful, as our friend Ian Malcolm reminds us. We can't confuse chaos, sensitivity to initial conditions, with what I'm just going to quote as real noise. So this is where the Gillespie algorithm comes in terms of simulation. It's very simple to simulate these sorts of master equations. There are really only 4 core steps in this, and involves computing these propensity functions and a cumulative intensity. And the idea is that we sample from some sort of distribution to understand when the next reaction fires and what the next reaction that fires is. Once we have that information, all we have to do is update our state x by some vector that describes the changes. So, for example, the time to the next reaction, you can think of each reaction as having its own internal clock. And these clocks go off at an exponential rate that is somehow related to the intensities for each of these reactions to fire. So you can see reaction 6 is much more likely to fire than reaction 5 because it has a higher propensity. And once we have that information, once we know which reaction fires next, all we have to do is sample according to how these propensities are weighed. And the takeaway from all this is that this approach might be useful. We might actually be able to use this to answer questions of biology because it's very easy to implement. And in fact, these types of algorithms have been around for decades. So this is where my package comes in. The idea behind BioSimulator.jl was to implement a modeling tool that strips away details about the math that you might not really need to worry about in modeling some sort of problem. It allows you to focus on just analyzing data and allows you to do this in a hopefully fast and easy to use fashion. Julia made this very easy to accomplish. The type system is especially helpful in being able to dispatch on different types and actually get down to writing performant code without worrying so much about having to maintain messy lines of code. As I mentioned, behind the scenes, we have a bunch of different types that handle the logic of computing these propensity functions, handling the logic of actually selecting reactions and understanding how to update the system, as well as some internal structures to maintain numerical errors, because these do pop up really quickly if you're not careful. So what does it do? It basically allows you to describe your model in a very simple and hopefully readable syntax to a user. As you can see here, you can define a simple network just by specifying what are the players or the species that appear in your network, and what are the rules that govern the dynamics of this network. So I have here an example with 2 species, X and Y, and the X species can duplicate itself by going through some birth event or reaction, and the Y species can annihilate X to effectively deplete the X population. Once you have your model description, you can just plug it into the simulate function, and from there carry out your simulations in a hopefully very fast fashion. We support a number of different algorithms, including the original Gillespie algorithm that I described, some other variations on the same idea, known as the first reaction method, next reaction method, an optimized version of Gillespie's method, and as well as one algorithm developed by my advisor, Dr. Maricel, called the step anticipation tall leaping. But there is a plan to hopefully add more algorithms to this in the future. And really what I want to stress from this is that these algorithms have a very simple structure. It's just one giant simulation loop, and all you have to do is plug in the logic to actually simulate this. And you can see here a very simple example from Gillespie's method. All you have to do is just carry out those 4 steps, and I apologize for the ugly print statements down there. And no presentation on a Julia package would be complete without some benchmarks. The benchmarks might be a little bit misleading in that Stochastic, which is written in C++, automatically selects a very efficient algorithm. So it's not, I tested this using complete default settings. If you tell Stochastic to use Gillespie's method, what is it going to do? If you tell my software to use Gillespie's method, what is it going to do? Stochastic has the nice feature that it allows you to select an optimized version, or it defaults to an optimized version based on some analysis that it does behind the scenes. And it also does this by utilizing several threads on your machine without you telling it to. So that's really nice. But even with all this, I'm able to get to very close performance as a mature C++ library. So the takeaway is that even though my implementation might not necessarily be optimal and have all these extra analysis features, it's very feasible to get towards that performance, if not better. And you can just look at these examples later on as to what exactly the benchmarks are doing. I have a little bit of time to go through one numerical example, and I'm going to choose the brush-slater cascade. The idea behind the brush-slater is that it's a system of coupled reactions, and the idea is that you have one species X that is catalyzed by some other species Y, but X itself is involved in the synthesis of Y, so it catalyzes itself in that sense. And I came across this paper with the brush-slater cascade that basically takes all of these little brush-slaters and couples them together to produce this very interesting system where normally if you try to model this in a deterministic setting, what you get is a fixed point under certain parameter regimes. But what you can see with this is that you actually start to see some noise further down the stages of the brush-slater. So as these reactions takes place, the noise is amplified through the biological or through the chemical network. So normally the fixed point would be somewhere around right here, 100, 100. So in the first brush-slater, there's no, like even though there are these deviations from this fixed point, you can still see that it's kind of hovering around that value. Whereas on the right side, further down this cascade, you see that you do in fact have oscillations. And going further up back here, you can see this a little bit clearly, there actually is a discernible pattern between these peaks and valleys as opposed to just random fluctuations. Okay, and I'd like to end my talk by thanking my advisors, Dr. Mary Sell and Dr. Kenneth Lang, for the support on this project. I'd like to also thank the Biomathematics Department, and thank you for the Carol Newton Travel Award, which funded my travel here. Thank you all for listening.